Welcome, NEET aspirants. This question is from the chapter Human Health and Diseases. So let's attempt the question now. Match question. Passive immunity. Passive immunity would be colostrum. D1. D1 is in option number 2 and 3. Next, cell mediated immunity. This is T lymphocytes. So C2. C2 and D1 is in option number 3. So we have the answer with us. Mark it on the OMR sheet. 199 is 3. And we are done with this question in 30 seconds. So next one, produce an army of proteins in response to pathogen. Army of proteins is immunoglobulins, antibodies, and it is produced by B lymphocyte. So A will be 3. And last one is innate immunity. So interferon, the cytokine barrier of interferon, comes in innate immunity. So B is 4. A3, B4, C2 and D1. So let's understand each and everything asked in this question. First one, passive immunity. Passive means when the immune system doesn't work to contribute to the immunity. We get ready-made antibodies. So when ready-made antibodies are directly given to protect the body against foreign agents, it is called passive immunity. Two examples are, so passive natural immunity is yellowish fluid called colostrum is secreted by mother during initial days of lactation. This colostrum is rich in antibodies, secretory antibody which is IgA, and it helps to protect the infant. And before birth, we call it fetus. So fetus is connected to mother via the placenta. So fetus receives some antibodies from their mother through placenta. So let's use this question as an opportunity to revise other categories of active immunity and passive immunity. So active immunity is when the immune system works to develop immunity and develop the antibodies and cytotoxic T lymphocytes. When ready-made antibodies are provided to body, then it comes under passive immunity. So natural examples we have seen, mother's antibody transferred via placenta and mother's antibody transferred via colostrum. This was asked in this question. And artificial passive immunity is, so in case of snake bite, antibodies against venom are given. Then viruses, the best example is rabies. So in case of dog bite, the injection which is given contains antibodies against rabies virus. Then anti-tetanus injections are given in case of injury due to rusted nails. Whereas in active immunity, immune system works to develop antibodies and T lymphocytes against the pathogen. Again, two subcategories in active immunity are natural, when natural infection occurs with microbes. That will come under natural active immunity. An example of artificial active immunity will be vaccination. So in vaccination, attenuated microbes are given. Attenuated means they are grown in culture and this way they lose their pathogenicity. So when attenuated microbes are given in vaccine, the body recognizes the pathogen, but their virulence is lowered. So their ability to produce disease is almost nil, but they are alive. And because they're alive, the immune system will recognize it, target it, and actively produce antibodies and CTLs against the pathogen. So it mimics natural infection, but it is given artificially. So that's why it comes under artificial active immunity. So you should have clarity in these 4 types of immunity. So we covered the first one, passive immunity. Next one will be cell mediated immune response. So cell is the T lymphocyte. So T lymphocytes mediate CMI, which is cell mediated immunity. And this is seen in organ transplant rejection reactions. So human heart, eye, liver, kidney, in all these cases, rejection reaction can occur if there is a mismatch and the recipient's immune system targets the transplanted organ. So rejection reaction occurs because the immune system is able to differentiate self antigen and non-self antigens. And this is primarily carried out by T lymphocytes. So T lymphocytes will match with cell mediated immunity. Then next one, B lymphocyte. B lymphocytes produce an army of proteins. These are antibodies, immunoglobulins, in response to pathogen into our blood to fight with them. These proteins are called antibodies. Antibodies are Y-shaped structure. So this will be the heavy chain and these two will be light chain. So this is a region which binds to the antigen. Okay, so since there are so many antigens, so this region of antibody will differ. Each antibody will have unique antigen binding site, but this region is called constant region. So supposing I have two different antibodies, one is against, let's say, HIV, and this one is Salmonella typhi. So which part of the antibody will be different since they are recognizing different antigens? This region, okay? This is called FAB region. Fragment antigen binding. So this structure. So both heavy and light chain, they create the antigen binding site. And two sites are available here. Similarly, this is the FAB for Salmonella. Now, if these two antibodies are belong to the class IgG, there this region will be common. This will be constant. The sequences of this region will be same because this region helps in interacting with other components of immune system. So antibodies are just like tags. They will specifically bind to the pathogen against which they are specific to. So their specificity is so strong, they can identify the antigen in 1,000 different antigens. So this is called the specificity of antibodies. And the constant region, it helps in activating other components of immune system like NK cells or complement, okay, or macrophages, which can engulf and destroy the pathogen. So this is how B lymphocytes work. Then different types of antibodies are IgA, IgM, IgE, and IgG. So IgM is produced in the beginning. So if you see IgM antibodies against any pathogen, that is indicative of a recent infection. And then slowly IgM is replaced by IgG. In body fluids, IgA is secreted. It is also called secretory antibody. And IgE are against protozoans and helminths. Okay, so this is also involved in the allergic reactions. So these are the four types of antibodies. Because these antibodies are found in blood, the response is also called humoral immune response. So humor indicates the blood, okay? So humoral immune response basically talks about antibody mediated immune response. So the main point which was asked in this question is match B lymphocytes with their ability to produce an army of proteins. So the last one is innate immunity and the option is interferon. So interferons are secreted by virus infected cells. They are a type of cytokine and this will protect the nearby non-infected cells from further viral infection. So this is a mode of communication within the cells. So innate immunity has 4 different levels. So in innate immunity, you need to remember 4 different categories. The first one is the physical barrier, the intact skin and mucosal lining This physically prevent entry of microorganisms. Physical barrier is the first line of defense. Then comes physiological barrier. Example is the acid in stomach. This also helps in eliminating a lot of microbial load which can come from food or water. Then third level would be the cellular response. Cells of innate immunity are NK cells and then neutrophils and macrophages. So they also help in destroying the pathogens. And the last category is asked in this question, interferon, the cytokine barrier. So cytokine, these are the messenger molecules which are released by the immune cells, and they help in communicating within each other and also with the other cells of the body. So these four categories you need to remember. So that's all about this question. Thank you so much for watching my video.